Of proteins. Protein structure starts with what we call the central dogma of molecular biology. And what this dogma states is that everything starts with DNA, and the message in DNA gets transcribed into RNA, and then that message in RNA gets translated into a protein. And ultimately, it's those proteins that are the workhorse of the cell, the workhorse of life. They're the things that do most of the things in our bodies. The central dogma was first discovered and set out by these esteemed scientists, Watson and Crick, and they're the ones that are famous for discovering the shape of DNA, the double helix, but they also did a lot of work later with DNA and help us to unearth this central dogma. And so over here we actually have a picture of the central dogma. So we can see here inside the nucleus, this is where DNA is at, and it's the message in DNA that gets transcribed into RNA. So here's our RNA molecule. That molecule then leaves the nucleus and goes to the ribosome, where it gets translated into a protein. And so proteins are made up of these structures called amino acids, and we string amino acids together to get a protein. So here we see our basic structure of an amino acid. It has three parts. It has an amino group, which is this nitrogen with some hydrogens. It has a carboxyl group, which can be a type of acid. Here it can actually donate a hydrogen ion. And then we have this variable group, the R group, and it's this R group right here that actually is what is going to determine what type of amino acid we have. There's 20 amino acids that we require. I think that there are 9 that we can't produce on our own, so we have to get them from our diet. But the different groups that go where that R group is are going to be what makes one amino acid different from another. Here we have what we think of as the 20 standard amino acids. And notice they're separated into groups. We have some amino acids that are nonpolar, and notice it's their R groups that make them nonpolar. We have some that are polar, and if you look at their R groups, they have elements about them that are going to make them polar. For example, this nitrogen here is going to be slightly negatively charged, so it'll actually act as a base. It'll sometimes pull positive hydrogen ion out of solution. So we've got some amino acids that are like bases, and some that are like acids, some that are polar, some that are nonpolar. But it's that R group that makes them different from one another. So how does this relate to protein structure? Well, there are 4 levels of protein structure, and we're going to look at each of them. The primary structure is simply the order of amino acids. So you'll notice here we have carboxyl groups and amino groups alternating, and then you can see we have the R groups here kind of alternating. And so what we really have right here in the primary structure is just a string of amino acids, one amino acid connected to another. And the central dogma tells us that the information that tells us what order to put those amino acids in comes from our DNA. The secondary structure is actually the interaction between the R groups of the amino acids in the primary structure. The tertiary structure is actually going to be interaction of secondary structures. So once the secondary structures form, we're going to have an interaction between them. And then the quaternary structure is going to be basically a conglomeration of multiple proteins put together. So for example, this protein is hemoglobin, and you can see it's actually got 1, 2, 3, 4 subgroups or subunits. In other words, 4 smaller proteins that come together to make it up. Here we have a picture of our primary structure. And so you'll notice what we just have is we have one amino acid bonded to another. So we've got like glycine, proline, threonine, glycine, threonine, glycine, glutamine, serine, lysine, etc., so forth and so on. Most proteins are somewhere between 200 and 2,000 or so amino acids. So we're talking very big molecules. Once this primary structure is formed at the ribosome, the R groups are going to start to interact with one another. Here are some possible ways that we could have interaction with the R groups. We can have, and actually we do have a lot of hydrogen bonding. So see, we have this slightly negative oxygen, which is going to be attracted to this slightly positive hydrogen. And so we'll get hydrogen bonding, and that will help hold our structure together. We can also have a very strong bond called a disulfide bridge. So some R groups will actually have a sulfhydryl group. It's one of our functional groups. And that will actually bond to another sulfhydryl group on the other side of the chain. We can have ionic bonding where we have actual transfer of electrons. And so we'll have ions. We'll have a negative ion and a positive ion, and those attract. We also have interaction of hydrophobic properties. We talked about how some of the amino acids have hydrophobic properties, and so all of the hydrophobic R groups are going to want to get together with one another, usually on the inside of a protein, and the hydrophilic will be on the outside, because outside the protein is lots of water in solution inside the cell, inside your body, inside the cells and the body cavities of living things. There's lots of water, so those hydrophobic regions want to get together. Let's talk about secondary structure. Secondary structure, remember, is just interactions of the primary structure. So we have two basic secondary structures. We have what we call the alpha helix, and that is just where we have it's a single-sided helix. It's not like a double helix in DNA, but it's just kind of like a swirling of the chain of amino acids. And then we can have what we call a beta sheet, which is just kind of like a solid sheet, and it's usually held together by lots of hydrogen bonds between the amino acids. And these are our secondary structures. So tertiary structure, we're going to see in just a second, is actually a combination of these secondary structures, the way they interact with one another. Here we can actually see in the secondary structure the hydrogen bonding that I was talking about, and this is actually an alpha helix. And so that is directly due to the primary structure. It's going to be the interaction of those R groups in the amino acids. Now, if we look down here, what we can see, this is tertiary structure. So notice we have several alpha helices that are interacting with one another, and there's going to be bonds between these that actually hold them together. And so now our protein is starting to take on some type of shape. The last level of structure for proteins is what we call quaternary structure, and that is simply several smaller protein subunits coming together to make one larger protein. So this protein subunit is going to come together with three others, and they are going to together make a very large and functional protein. Most proteins that are functional in biological systems are a conglomeration of multiple protein subunits and therefore have quaternary structure. Here are a couple of examples of some proteins that have quaternary structure. Notice we can see in these proteins the secondary structures, the alpha helices, and we can even see we might be able to see some beta sheets kind of right here where it's not really, doesn't look very swirly. And so this right here is hemoglobin, and so we can see it has four different subunits that have come together to make one protein. We now are able to actually elucidate the structure of various proteins and study them and even build them using biotechnology, using bacteria to actually create and manufacture in mass quantity proteins. So if you look at this graph right here, what it shows you is way back in the 1970s. Now this is, about 20 years, 15, 20 years after Watson and Crick discovered the central dogma and the shape of DNA, and we didn't really know the structure of very many proteins. And so what this graph shows is how over time our knowledge of protein structure has increased exponentially. So way over here in 2010, we currently know around 70,000 different proteins and what their exact structure is. And we're able to replicate those in a lab. We can draw them out. We can use computer-generated images to actually explore them. And notice that our knowledge has been exponential. In other words, over the years, the more we learn, the more we're capable of learning. And so really, it's only been in the last 15 years or so, 15, 20 years, that our knowledge of protein structure has really taken off. So why is that a big deal? Why does it matter that we know about protein structure? Well, it turns out that it's actually the structure or the shape of a protein that determines what it does, and this is best exemplified by enzymes and substrates. If we we think about our basic enzyme, it has an active site, and there's only one particular substrate or molecule that fits in that active site. In this particular illustration, it is the disaccharide sucrose. So here we have glucose, which is a 6-carbon sugar, a hexose, and we have a pentose, a 5-carbon sugar, which is fructose, and they're bonded together in a disaccharide. And this particular enzyme breaks them down, and it is the fact that it fits perfectly with that molecule like a lock and a key, and it is actually able to add water. So we're using hydrolysis here to break the bond between them, and we can break them down into 2 monosaccharides. So by knowing the exact structure and shape of a protein, that gives us an idea of what that protein does. So let's do a real quick recap. Central dogma tells us that everything starts with DNA. The code in DNA gets transcribed into RNA and translated into a protein. So we start out with DNA in the nucleus, we carry the message out to the ribosome, and the ribosome builds us a protein. Once we have built this protein, the way that it actually folds and changes shape is we start out with our primary structure, which is just a sequence of amino acids that was determined by the DNA. The primary structure, the R groups interact to form secondary structures like alpha helices and beta sheets. The alpha helices and beta sheets of the secondary structure combine and interact with one another to make tertiary structure. And then we can actually put several proteins together, several protein subunits together to make one large protein, which is our quaternary structure.